So I'm going to go ahead and start to record now. So happy Tuesday and congratulations on making it through half of the class. So we're in week 4, which means that we're halfway done with the semester. And I was just looking at everybody's grades. I looked at them over the weekend, but then I was looking at them today too. And I adjusted one of the columns in the full grade center just so I know you guys see it in a different way. You see like your grades, but I adjusted one of the columns so that you could see like there's your total column. So and so there's like, it looks kind of ridiculous. You're like, oh, I have 400 points, right? But to make that meaningful, I put an option so you could see what the letter grade associated with that is. And so overall, the class is doing fantastic. Right now, everybody is passing the class. So you're doing really well. I forgot last week to review the performance of how you had done as a class on exam 2. So I wanted to start with that. So let me go ahead and share hold on, let me make sure that I'm not showing any private information first. Okay, so show this and then sheet 2. Okay, so let me share Excel. Okay, so what you should see right now is just a graph of the overall class performance for exam 1 and exam 2. The blue is exam 1, so you can see that kind of bell-shaped curve with the average being kind of in this B range. But with the exam 2, you can see it's in the orange, and so there was a shift towards higher grades, and there were no D's and no failing grades. So it was a great performance. So for this sheet just shows all of the grades, and I'm really just showing it to you so that I wanted to highlight the average grades. So from exam 1, let's see, this is exam 1. It's the average was an 81.8. And so for exam 2, the average is a little higher. It's actually closer to an A. So exam 2 average was an 89. Exam 3 is open now. I see some of you have completed it already. So that's just let's see if you guys continue with this really great performance. So we're done with that. I just didn't want to leave that hanging for you. So I did check the questions on exam 2. There was one question that I noticed that it was just kind of like an unusual amount of students that had gotten it wrong, and it turned out that I had chosen the wrong answer in the answer key, so I corrected that. So at some point, probably on Thursday, you might have noticed that your grade went up a little bit. I think everybody chose. This is why I was like, why did everybody get this question wrong? And the truth was that you hadn't gotten it wrong. I had put the wrong answer in. So I corrected that for you. So you would have noticed a little bit increase in your grade. That didn't really affect the average. It was really high to begin with anyways. So let me go ahead and review what we have going on for today. All right, sharing Google Chrome content. All right, okay, so you should be seeing our Blackboard page now. So half of the class is done. We're in week 4, and we're looking now at our larger, more multicellular organisms, starting with your fungi. And then from your fungi, we'll be looking at the animal group. So we also have our introduction to animal diversity today as well. And then on Thursday, we'll introduce your protostomes, which are primarily the majority of your invertebrate organisms. And the next week, we'll start with your vertebrates and the final invert groups. So for this week, there will be 3 SmartBook assignments. 2 of them are assigned today for chapter 31 and 32. There'll be another one that's actually live already, but another one for chapter 33 on Thursday. All of those are due on Monday evening, and I've been trying to send out reminders through Blackboard on Monday, just to if you hadn't turned them in, and it's been working. I have 100% participation on those, and you guys are getting things in on time, which is fantastic because it means you're not losing unnecessary points. So keeping track of those due dates means that you're earning everything that you possibly can. So for today, there is a participation assignment. There'll be one again on Thursday. So let's click in here. I'll just show you. So exam 3 is up and available, became available at 9 this morning. Your SmartBook assignments are there. And then we have an assortment of different videos, video lectures for chapter 31 and for chapter 32. And for chapter 32 in particular, I tried to break these into like each of the different segments so that they are accessible. The information that we're introducing in chapter 32, we will reintroduce again at the very end of the semester. This sets the stage for you to understand how your protostomes and your deuterostomes are different, and we're introducing some terminology that helps us differentiate the organisms and provide some of the characteristics about each of the organisms. But we will talk a little bit more about animal development towards the end of the semester, which is kind of a precursor to if you're at all interested in taking a developmental biology course, or are interested in things like stem cell biology, or it sets the stage for higher level courses. So for this lecture, there's a number of videos, and most of them are just kind of interesting videos related to the organisms that we're looking at. So I have, we introduced the sponges, which are the most simple animal group, and they lack tissues, and they lack tissues, and they lack symmetry. And so I have these 2 videos of them. I mean, honestly, if you just go, snorkeling in the water and you come across sponges, you're just like, oh, sponge, right? But so these actually are some relatively interesting things about your sponges. If you ever take marine zoology with Dr. Middlebrooks, he makes all invertebrates fascinating. You will like the class. But one of the field trips that he regularly does is to visit, there's the sponge docks, which are up in Tarpon Springs, where they go and collect. I mean, the field trip, I don't think you are collecting sponges, but that area, they actually harvest sponges in that area. And then I also include some videos of different gelatinous organisms. So as a personal note, I love jellyfish. The way that I became interested in being a scientist was when I was around your age. I was an undergraduate, I was a sophomore in college. I started doing research with a faculty member, kind of like me, and the project that I started working on was with ctenophores, which are a non-stinging type of jellyfish, and I was looking at the feeding behavior of them. So I have always found them fascinating, and have kind of done a little work with these ctenophores kind of for whatever, 20-something years. I also really like jellyfish too. Anything plankton, I like plankton. So I can't get away from trying to share interesting videos about different types of gelatinous organisms. So those are there so that maybe you love them as much as I do. I also just want to reiterate that we have the brochure project coming up, and not this Friday, but the next Friday. Talk to me if you have ideas about what you want to present, and we can go over that. I'm happy to, if you're confused about anything, I'm happy. I love thinking about biology, so that would be enjoyable for me. All right, so let's see, stop sharing that. All right, so the last couple of things that I wanted to talk about today were to go over the common misconceptions. And so before I start going over the misconceptions, I wanted to do the poll, because I like this poll. Gets you to interact. So I wanted to go over this because I think that will lead into, you know, opening up some of the common misconceptions for me. So, all right, so let me find today's poll. All right, the first one is fungi. So I'm going to launch this. We'll do about 2 minutes for you to go through. I tried to make them all true and false. I found that those allows you to kind of answer them more quickly, and they're less confusing. All right. All right, we're going to give it about 15 more seconds. Five more seconds if you want to answer. Okay. All right, so let's look at this. So you should now be able to see the results. Okay, so let me put this over here so I can see. Oh, very interesting. So for the first question, it's 50-50. That's really interesting. Okay, so fungi are more closely related to plants than animals. All right, so that isn't supposed to be a trick question. I mean, but I did expect this to be something that you're unsure of. So in fact, fungi are more closely related to animals than they are to plants. What? But your fungi are part of the same supergroup that animals are. They're part of the Opisthecanta, whereas your plants are in a different supergroup, the Archaeplastida, which they're more closely related to your green algae. So this is actually false. Fungi are not more closely related to plants than animals. But if you look at a fungus, right, they don't look a lot like animals, right? But they do share more characteristics with animals than they do with plants. So that is a common misconception. All right, so fungi do not produce gametes. All right, so interestingly, these organisms do not produce gametes. So the correct answer is true. They generally produce spores as their form of reproduction. So there isn't you'll look at the life cycle in today's video lectures, but yeah, there are no gametes that are produced. And though they do have sexual reproduction, there's just a fusion process where your two hyphae meet and kind of fuse together. It's different than what we see in either the animals or the plants. They are very unique. But yeah, no gametes are produced. All right, so like I just said, fungi do have sex. So we know they have sex, but they do not have male or female genders. So the majority of you answered true, and that is actually true. So interestingly, there isn't genders. What we refer to them as are positive and negative strains. So there are different hyphae that will tend to be attracted to one another through a release of chemicals, and they'll grow to one another. But there isn't just a male or a female. It's just they're just different strains that are attracted to one another. And then the last one, in sexual reproduction, the cytoplasm and the nuclei of the mates fuse simultaneously. Okay, so when we have, or animals have reproduction, you have your sperm and your egg, and they fuse, and both you end up with your nuclei and your cytoplasm kind of fusing at the same time. So that's generally what happens. With your fungi, we're going to introduce a different process in their reproductive cycle, where first their cytoplasm fuses, and that's called plasmogamy. And then later, at some point, it could be weeks, it could be months, it could be years, at some point later, the nuclei fuse. So at certain points, you could sample fungi and there will be two separate nuclei in the cytoplasm. So they do not fuse simultaneously. So this answer is actually false. It's possible that there are other organisms like this, but this is the main kingdom in which these two processes occur separately. Fusion of the cytoplasm, plasmogamy, occurs before fusion of the nuclei, which is karyogamy. And so I wanted to introduce that first. I'm going to stop sharing now, because it is pretty unique for these organisms, and I wanted you to pay attention to it when you're looking at that. All right, so with our fungi misconceptions, so one of the things that this figure has allowed us to introduce is that when you're talking about your fungi, they're not all just food products, right? Fungi are actually a lot more diverse than just the portobello mushrooms that you find in the grocery store or what you add to your salad. The majority of fungi are actually existing in this hyphal form, these network of these filaments, and they are ecologically essential. So in this respect here, it's serving as a decomposer. And for life to exist, we need this recycling of organic material, the carbon, to be re-released for use by other organisms. And also, for food production, fungi and plants have a symbiotic relationship in a lot of sense, in that fungi and plants kind of co-evolved. So without fungi, our plants would not be successful either. So the fungi have actually enhanced the success of plants. Your fungi were colonized the land first and then made the way possible for your plants. All right, so the next confusing one was related to a lot of the questions that I introduced here. There is a lot of confusion for students with the life cycle of the fungi. It's not as complicated as alternation of generations, but the main thing that I tried to introduce too is that they have this separate fusion of your cytoplasm and separate fusion of your nuclei. So pay attention to that when you're looking at the life cycles. Another interesting feature of these organisms is that they have a very short-lived diploid phase. Most of your fungi exist in a haploid phase. And so there's a really short-lived stage in which it's diploid, and when that occurs, the process of meiosis occurs relatively quickly in order to restore the haploid form within these organisms. Once again, as with your plants, we see a production of spores, and the spores are not related to sexual reproduction. It's an asexual process in which your spores are produced. And then also to reemphasize, there are no males and females. That's not relevant with your fungi. They exist and they mate, but there isn't a separate male or a separate female. And the other interesting fact about your fungi is that these are multicellular eukaryotic organisms, but they don't have tissues and they don't have organs, right? So when different processes happen, they're not happening in an organ system. All right, and then I think that was it for your fungi. Okay, so let's now talk a little bit about the animal diversity section. I'm gonna launch the animal development poll. This one only has 3 questions. Okay, so you should be able to see that now. All right, so we'll take about a minute and a half. It seems like most people are done in that period of time for you to answer those couple of questions. And you may not recognize these terms, and that is okay. All of these will be explained to you in the video lectures. My husband's walking behind me, making a lot of noises right now. Fantastic. All right, we'll give you guys about 15 more seconds. Everybody answered. Oh my goodness, this is a miracle. All right, I'm gonna close this now. All right, so we'll share the results. Dun, da, da. All right, this is fantastic. You guys, as a whole, the correct answer was true for the first one. Animals are ingestive heterotrophs. This is kind of how we often identify your animals. Animals are multicellular organisms that are mobile, most are mobile, and that they are heterotrophs, but they're ingestive heterotrophs. And your fungi, while also heterotrophs, absorb their organic material rather than ingest it. So the correct answer is true for that one. The second question here is something that I want you to pay attention to. So we'll introduce the concept of a coelom, which is a body cavity. And with your triploblastic animals, not all of your triploblastic animals have a coelom. So the correct answer is false. The ones that lack a coelom, those are organisms that we call acoelomate organisms, and one of the examples we'll look at are your planarians. All right, and then the last one, this will be discussed when we talk about what the differences are between your protostomes and your deuterostomes. And so it says in both protostome and deuterostome embryos, the blastopore develops into the mouth, and this is false. So for your protostomes, proto means first, stome means mouth. So for those organisms, the blastopore becomes the mouth. But for your deuterostomes, which we'll talk about in more detail next week, for those, the blastopore develops into the anus. So deutero means second, so the mouth forms second. So the blastopore develops into the anus, and then the mouth forms second. So in only the protostomes, the blastopore becomes the mouth. All right. All right, so I'm gonna close that. All right, so I don't have a huge, there's not a lot of misconceptions here. More will come when we look at the protostomes and deuterostomes in more detail. But the things that I want you to pay attention to are related to how we distinguish the protostomes and the deuterostomes. I'll introduce words like spiral and radial cleavage and coelomate and pseudocoelomate. So I try to include little video clips of what that means or how those processes of cleavage work. If those are unclear, Google and YouTube are your friends. There are a lot of other additional videos. I did not want to overburden you with videos on how all of those processes work, but there's more tutorials on cleavage and how those processes work. And also, I'd also recommend I frequently recommend drawing out different concepts, but particularly when you're looking at your triploblastic organisms, if you try to draw out where the different tissue layers are so that you can visualize where the coelom is and what type of tissue surrounds the coelom, you'll better understand the difference between a pseudocoelomate and a coelomate. The acoelomates are pretty easy because there's no body cavity in those organisms. But so like I said, I recommend drawing. It helps with life cycles. It helps with different layers of tissues and bodies. Drawing things out will also help you if you're going in the in any field, but particularly pre-health fields, because you'll need to know where different organs are and different tissue layers are, or different muscle systems. So being able to connect that visually will very much help you. And then the last thing I wanted to mention is we're starting to talk about different animal groups, and certain words are not interchangeable. So just pay attention to that. So not all organisms are animals. So plants are organisms, fungi are organisms. So animals are a type of organism. And not all animals are vertebrates or mammals, right? We tend to be very human-centric and think we are the epitome of all animals, right? But most of the animals that we're gonna talk about are actually invertebrate organisms, so they lack a backbone. They seem more simple than humans do. So just remember that we're talking about different levels of within the kingdom Animalia, right? So kingdom Animalia is really broad, and then we're going to talk about the specific groups in more detail. All right, so that is everything that I needed to share with you so that you could be better prepared to be successful. I wish you the best of luck as you complete exam 3 today. If you have any questions, then please email me or visit me in Zoom, or we can set up a meeting for that. I would love to talk to you guys in more detail, but other than that, you are free to go for today, and I hope everything has made sense so far. All right, have a great day.
Thank you.
You're welcome.
Thank you.
Bye.